Hello everyone, my name is Dr. Shravani, and from today onwards, I have started the NEET revision session, wherein the first chapter I have taken is biomolecules. So if you talk about the weightage of this chapter, in 2024, there were 4 questions asked. In 2023, again, 4 questions. In 22, it was 5 questions. In 2021, it was 7 questions. So on an average, we can expect around 4 to 5 questions in NEET from this chapter. And if you talk about the weightage, it forms almost 10% of the zoology question paper for the NEET exam. Okay. So with this, we can say this chapter is very important, isn't it? Now, so before starting any chapter, usually I talk about the contents of the chapter so that like when you go through the contents of the chapter, it will tell you what exactly are the topics we are going to cover and it will give you the brief idea of the chapter. So first we'll start with the contents and then we'll go into the details of the chapter. Coming to the contents, first, we'll be talking about the meaning of biomolecules, what is the meaning and everything. Then we'll talk about the types of biomolecules. So basically, we have 2 types, biomicromolecules and biomacromolecules. So what are the types, on what basis we have classified these biomolecules, that we'll be discussing here. And then we'll be talking how we have separated the biomolecules into micro and macromolecules. On what basis? Okay. So for this, we have to do some certain experiment, which we call it as chemical analysis. So we'll be basically analyzing. Analyzing is studying or observing the chemicals which are present in the living tissue. So there, after doing the chemical analysis, we get certain chemicals, that is certain compounds, and when you analyze them, there we can get biomicro and biomacromolecules. After doing chemical analysis, we'll do one more experiment, which we call it as ash analysis. So here we get something called ash, and this ash will be studied or observed. So basically, this ash analysis will give us the inorganic components of the living organism. So inorganic components includes your calcium, magnesium, potassium, all those things. So these will be observed when we do the ash analysis. And when you do the chemical analysis here, basically we'll find what are the organic molecules. Apart from organic molecules, we also see some inorganic components. Not all, but few inorganic components we can find in chemical analysis, but in ash analysis, definitely it is only the inorganic component what we get. Once we finish all the introduction part of the chapter, then we'll go into the details of the 4 biomolecules which are present, which are given in your NCERT. The first one will be carbohydrates, which are otherwise called as the polysaccharides, or we can say the sugars, all those things. And then we'll be talking about the proteins, lipids, which are nothing but the fats. And then we'll be talking about the nucleic acids, which are nothing but DNA and RNA. Once we complete all the 4 biomolecules, that is carbohydrates, proteins, lipids, and nucleic acids, then we'll be talking something called metabolism and living state, which is very small topic. After completing this, we'll be going into the enzymes. Now, enzymes is again very important from exam point of view. Here, we'll be discussing what do we mean by enzymes, how do they work, that means like how do they increase the rate of metabolic reactions or the chemical reactions, and what are the factors responsible for the enzyme activity. And after that, we'll be discussing the classification of the enzymes, what are the different types of enzymes. And at the end of the chapter, we'll be discussing about cofactors. Now, cofactors is also very, very important from exam point of view. Definitely one question will be asked from the cofactor concept. Okay. So these are the contents of this chapter. Now we'll start with the details. So coming to the first topic of this chapter, as I have told, we are going to learn about the meaning of the term called biomolecule. So what do we mean by biomolecule? In your NCERT, they say that all the carbon compounds which are present in the living cell are considered as biomolecules. So they have stressed on the carbon compounds present in the living cell. So does that mean carbon dioxide, carbon monoxide are also the biomolecules? Answer is no. Carbon dioxide, carbon monoxide, whenever they are present in the cell, they are not the biomolecules. Okay, why they are not the biomolecules? Because they are the inorganic components. Okay, so they are inorganic components. So basically, when you go in depth of the biomolecules, they are actually the organic carbon compounds. So whatever organic carbon compounds are present in our body, those compounds are considered as biomolecules, but not the inorganic ones. Inorganic includes your carbon dioxide, carbon monoxide, calcium, magnesium, phosphorus, potassium, all those things. Okay. Then what exactly are the examples of your biomolecules? It will include your carbohydrates, proteins, then we have amino acids, then we have nucleic acids, then nucleotides, and comes your lipids. Okay, so we have carbohydrates, proteins, amino acids, lipids, nucleic acids. All these things forms your biomolecules, but not the inorganic components like carbon monoxide, carbon dioxide, magnesium, calcium, sodium, potassium. Okay, these are not the biomolecules. Now, coming to the next topic, the types of biomolecules. So here we have types of biomolecules. Basically, biomolecules are of two types, biomicromolecules and biomacromolecules. What do we mean by the term micro? They are small. Macro means large. So here, the biomolecules which are present in our body will be classified based on their molecular weight. If the molecular weight is less than 1000 daltons, around 18 to 800 daltons, such molecules we call it as biomicromolecules. On the other hand, if the molecular weight is more than 1000 daltons, then those molecules we consider as biomacromolecules. Now coming to the examples. See, here you have to apply certain logic. Whatever polymers we see, for example, the polysaccharides, okay, polysaccharides, then we have proteins, nucleic acids. All these are the polymers. Polymers are nothing but chain of monomers. So all the polymers which are formed by the chain of monomers, all these form your biomacromolecule because the size of this molecule will be very, very big. If say this is your monomers, so we have so many monomers joined with certain bonds to form your polymers. So these polymers will be macro, they will be larger in size. Hence, they are considered as biomacromolecules. On the other hand, whatever monomers we have, which are forming these polymers, those monomers will be considered as biomicromolecules. For example, if you consider polysaccharides. So polysaccharides are made up of monosaccharides. So this will be your monomer. Proteins are formed by amino acids and nucleic acids are formed by nucleotides. Nucleic acids include your DNA and RNA. All right. So polysaccharides are formed by monosaccharides. Amino acids are formed by proteins. Amino acids forms proteins and nucleotides will form your nucleic acid. Now, all these components put together, we call it as monomers. So, logically, the polymer component which are present in our body will be considered as biomacromolecules and the monomers will be considered as biomicromolecules. Now, if you look at this list, we are left with lipids. So, what about lipids then? Lipids are neither monomer nor it is polymer. Then what is lipid? Lipids are complex molecules. Okay, so lipids, if you talk, they are neither monomers nor polymers. They are simply complex molecules, and these complex molecules have got the molecular formula or molecular weight less than 1000 daltons. So if the molecular weight is less than 1000 daltons, then such molecules we consider it under biomicromolecules. So these are the two different types of biomolecules which are present in our body, micro and macromolecules, wherein polymers will be considered as macromolecules, monomers will be considered as micromolecules. And if you talk about lipids, they are neither monomers nor polymers. They are complex molecules, which are also a type of biomicromolecules. Now, the next topic, what we are going to discuss is how to separate this biomicro and biomacromolecules. So as I've told, we do something called chemical analysis, with which we will be able to tell which of the molecules are macro and micro. Okay. Now, coming to the experiment, what we have to do here is you have to take a living tissue. Now, this living tissue can be a vegetable if it is a plant-based, and it can be, if you want to take any animal part, you can take a piece of liver or any other organ. Okay, so if you don't want to touch the animal parts, simply you can take a vegetable or a leaf. So you have to take this, and this living tissue you have to put in a mortar. Now this is a mortar. What do you mean by mortar? Mortar is, like a grinder, and we have a pestle. So in your, like mamas will be grinding the chutneys and everything. So that kind, the vessel, what we use is called mortar. So put that living tissue in the mortar. Along with this living tissue, you have to put something called trichloroacetic acid. Very, very important. And the molecular formula of this trichloroacetic acid is also very important in the exam. Usually the questions will be asked like, what is the chemical which we use during chemical analysis along with the living tissue? So answer will be trichloroacetic acid, that everyone knows. But there, they will confuse you with the formula here. So as we know, trichloroacetic acid, say this is carbon, to which three chlorides are attached like this, hence trichloro, and here we have a acid. So it is trichloroacetic acid. So Cl3, C and COOH. Okay, Cl3, COOH. So in the exam, they will tend to confuse you with, say, Cl3, C, then again C, then COOH. So like this, they want to confuse you. So you should not get confused. Please try to remember the formula also for trichloroacetic acid. Yeah. Now again, coming to the experiment. So take a living tissue, put that in a mortar. Along with that, put the trichloroacetic acid, and then you grind it nicely. What do we get after grinding? We get a thick paste, and that thick paste, we call it as slurry. Clear? Now, what you have to do with this slurry is you have to filter this slurry. Say, for example, this is your, take a test tube. Over the test tube, you put a cotton or a cheesecloth. Say this is your cotton or a cheesecloth, and then you strain it. Clear? Whatever thick paste you have got, the slurry you have got, then you have to strain it along with the cheesecloth. Now what happens when you are straining it? The things which got dissolved in the acid, that is trichloroacetic acid, will be present here, isn't it? They'll get filtered and it forms something called filtrate. So the part of the molecules which got dissolved in the acid will be filtered. We call it as filtrate. Otherwise, it is also called acid soluble fraction or acid soluble pool. And the one which did not get filtered will be retained on this cheesecloth, and this part we call it as retentate. Otherwise, acid insoluble pool. So these are the two fractions, pool, or we can say fraction. So these are the two things what we get once you strain it. Acid soluble and acid insoluble pool. Now what we have to do is we have to analyze, we have to study the components of these two things, filtrate as well as retentate. Okay, so when you study the components of filtrate and retentate, what are the things we get? Nothing but what we have just read now. So in filtrate, we see something called biomicromolecules. So in filtrate, we will see biomicromolecules Along with biomicromolecules, we also see something called inorganic components. Few inorganic components like your sulfates or phosphates. Okay, so we get biomicromolecules plus inorganic components will be seen in filtrate. Biomicro are the monomer part, okay, which includes your monosaccharides. Then we have the nucleotides and the amino acids. All these will be biomicromolecules. If you talk about retentate, what do we get in retentate? Here we see the biomacromolecules. Since they are very bigger in size, their weight is very large, so these cannot get dissolved in the acid and they will get retained on the cheesecloth. So we get biomacromolecules on the cheesecloth. Along with this, very important, we get lipids also. Okay, we get lipids also in the retentate. So here what you have to understand is lipids, just now we have read that it is biomicromolecule. Isn't it? Lipids are biomicromolecule because their molecular weight is less than 1000 daltons. So even though the lipids are biomicromolecules, still these lipids are found along with the biomacromolecules in the retentate. Now, what is the reason behind it? That is again very important. So for that, if you talk about the reason, okay, so next one, why lipids come in retentate even though their weight is less than 1000 daltons. That means even though they are biomicromolecules, why do we see lipids and with the retentate along with the biomacromolecules? So here the answer is very simple. See, when you are grinding the tissue, whatever it is, whether it is leaf or liver, when you are grinding the tissue, tissue is nothing but made up of cells. Now each cell, if you consider each cell, it is made up of cell membrane. It is made up of cell membrane, and inside this cell membrane, we have so many cell organelles. For example, if it is plant, we can have chloroplast, mitochondria, ribosomes. All these are different types of cell organelles. Similarly, animals also have got cell organelles. So let me write down this cell organelles. So cell is made up of cell membrane and cell organelles. And this cell membrane and cell organelles, the envelope, the outermost covering is basically made up of a lipid layer. Okay. And if you consider cell membrane particularly, this lipid layer is made up of two. So we have two lipid layers, which say for example like this, we have two lipid layers. This in detail we'll be studying in the chapter called Cell, the Unit of Life. Okay. So here, if you consider the cell membrane structure, it is made up of two layers. This we call it as bilipid layer. Bilipid layer. And this lipid layer, individual component, if you consider, say this is the individual component, which is nothing but phospholipid. So basically, the entire cell membrane is made up of phospholipids, which are arranged one after the other, just like how I have drawn. So each component is made up of phospholipid, and this each phospholipid is one lipid, what we have. Clear? So what happens when you are crushing the tissue, when you are grinding the tissue, basically you are fragmenting the cell membrane. So cell membrane is broken down into several fragments like this. Clear? And each fragment is made up of something like this. Just now I have shown you. So each membrane, each fragment is made up of several phospholipids. Now what happens? This phospholipids, as you have already, you might have read in Cell, the Unit of Life, it has got two parts. One will be hydrophobic and hydrophilic. Okay, so this head part, we call it as hydrophilic part. So this is hydrophilic, and this tails, the two tails of phospholipid are actually hydrophobic. So hydrophilic are water loving and hydrophobic are water hating. So in order to prevent the contact with the water, what happens to this fragment? They will again form a very small, bubble-like structure. For example, if this is the fragment, They say these are the fragments of the cell membrane. After crushing, this fragment will form a vesicle-like structure, something like this. So these are called as vesicle. And each vesicle, if you consider the structure of each vesicle, it will be something like this. So all the phospholipids are arranged to form a bubble-like structure. Clear? So now if you consider the entire vesicle, it is made up of several phospholipids. This is one, this is second, third, fourth. So we have so many phospholipids in each vesicle. Clear? And since we have so many phospholipids, obviously the weight of the lipid there is very big. Okay. So that is the reason why this lipid, after grinding, the fragments of the cell membrane or the other cell organelles, the membranes of other cell organelles, they will actually form the fragments, and each fragment will form a vesicle like structure. And these vesicles, they have larger size and larger weight. So they will get retained on the top. That is, in the cheesecloth itself, the vesicles will get retained. They are not getting filtered. Okay. When we can see the lipids in the filtrate, now this is not possible, but I'm telling you, when is the chance? If you could have broken the cell membrane into its individual phospholipid component. For example, if you could separate each and every phospholipid here, then we might have seen this phospholipid, each phospholipid inside the filtrate. But that is not possible with this experiment. Hence, we see the vesicles in the retentate only. Clear? So that is the reason why lipids, even though they are biomicromolecules whose molecular weight is less than 1000 daltons, still they are found along with the biomacromolecules in the retentate part, because once you grind the tissue, the fragments of the cell membrane, they are not broken into individual component of the lipid. Instead, we have the group of lipids whose molecular weight and size is very large, just like your biomacromolecules, and hence they get retained over the cheesecloth. This is the answer for that question. So this is all about our chemical analysis, which will give us an idea about the types of biomolecules, wherein we can classify biomolecules into biomicro and macromolecules. Now the next topic is ash analysis. Now, what do you mean by ash analysis? As I have already told you, ash analysis is done to study the inorganic component in the living organism. For this, what we have to do, you have to take a tissue again, a living tissue. Now, this tissue has to be weighted. So, living tissue again, you can take a vegetable piece or a leaf if it is a plant-based, or if it is animal, you take a piece of liver or any other organ. So, you take that tissue. Now, this tissue has to be weighted. Okay, weigh it. Once you weigh it, we get the wet weight of this tissue. Clear? After getting the wet weight, what you have to do is you have to dry it. So, you dry that tissue. Again, you take the weight and we get the dry weight. Now, after drying it completely, okay, so drying ensures that the water component has lost in it. After it is completely dried, you have to burn it completely in the presence of oxygen. Okay, so in the presence of oxygen, that means in the open air, you have to burn that tissue completely after drying. So when you burn it completely, what actually happens is whatever organic carbon compounds are there, right? Whatever carbon compounds are there in that tissue, they will get oxidized to form certain gases. What are those gases? It can be CO2 and water vapor. So these are the gases, carbon dioxide and water vapor. In this form, whatever organic components are there, whatever basically the carbon compounds, whatever carbon compounds are there, that will be lost in the form of carbon dioxide and water. And what we are left with is called ash. Yeah. So when you burn any living tissue in the presence of oxygen, that is in the open air, when you burn it, then we get the ash. Now this ash should be analyzed. Analyzed means studied. Clear? So what are the components when we do the ash analysis? We get all the inorganic components. Clear? Now what are the inorganic components we can get? We have sodium, potassium, calcium, magnesium, all those things. If you talk about compounds, it could be sodium chloride, calcium carbonate, phosphates, sulfates. So these are the different types of inorganic components we get when we do the ash analysis. So few points you have to remember here, that when you perform ash analysis, you get completely inorganic component. But when you do the chemical analysis, there are chances of getting both organic and inorganic components. This slight difference you have to remember. Now, this is all about the introduction part of this chapter. Now we'll start with the main topics, that is the biomolecules. So here the first biomolecule, okay, so before that we have a few tables which are very important from exam point of view. So the first table which is mentioned in your textbook is they have done the comparison between the elements between the Earth's crust and the human body. See, we know that whatever things are present on the Earth's crust, that is hydrogen, carbon, oxygen, nitrogen, everything are present in the organisms also. So same elements are present both in Earth's crust as well as a living organism. But if you consider the percentage of these elements, the percentage will vary. In the Earth's crust, say for example, if you consider oxygen, the percentage of oxygen on the Earth's crust will be 46%, whereas in human body it is 65. So it is very high. On the other hand, if you consider carbon, so carbon percentage on the Earth's crust is only 0.03 percentage, whereas in human or in any other living tissue, it is around 18 percentage. So it is very high. So even though the elements are same in both Earth's crust and the human body, but the percentage is different. Clear? So here, usually the questions are asked based on the percentage of human body, and they can ask you in order to arrange in ascending or descending order. So here I have written the descending order. That means from highest percentage to the lowest. For example, here oxygen, if you consider, we have 65% in the human body, and silicon is negligible. So it is negligible. So this descending order, or you can learn in ascending order, both are fine, but you have to learn because this is asked in the exam. So please don't leave this table, very important from the exam point of view. So you can make some tricks to remember this. Now I can give you one trick to remember this. The trick goes like oxygen key car. I'll write it so that it will be clear for you. So it is like oxygen की कार नगेश की है, एंड उस कार में सुमा एंड सोनम, मैगी खाने, सिलिकॉन वैली गए थे. So this is the trick you can use to remember the descending order. So here it goes like this. Oxygen ki car will be your carbon, Nagesh will be nitrogen, ki again will be calcium, hai will be hydrogen. Then comes Suma, sulfur, Sonam. So here I have written for sodium, Na, please don't get confused here. Sonam for sodium. Then magnesium, Maggi, and then silicon is for the silicon as it is. So you can use this trick, or otherwise you can use your own trick, but please try to remember the order here. So again, very important, and try to remember the values also. Next, we have one more table. Now this table shows us the composition of the cell. For example, say this is our cells with the cell membrane and the cytoplasm in it. Say we have cytoplasm, nucleus, and other cell organelles. So here they are telling the composition, the percentage of the composition of the components. For example, we know most of the body, in our body, we have water, isn't it? More than 75% of our body is made up of water. So same way, you can remember water is the highest percentage, 70 to 90% of our body or any cell is made up of water. Then comes protein. So first will be water, then the cell is made up of proteins. And then very, very important, it will be nucleic acid. So please don't get confused here. After proteins, it will be nucleic acid, and then comes your carbohydrates, lipids, ions. Usually what we think, it will be proteins, carbohydrates, lipids, and nucleic acids. But that is not the case. You have to remember, nucleic acids are more. Nucleic acids are nothing but your DNA and RNA. Okay. So DNA and RNA are more than your carbohydrates, then comes lipids and ions. And please try to remember the values also. So this is all about the tables, the first two very, very important tables. Then we'll be talking about something called metabolites. Now, what do you mean by metabolites here? Any components or any products, what we get after a chemical reactions are called metabolites. Clear? So these metabolites in our body are of two types. We have primary metabolites and secondary metabolites. Primary metabolites are the biomolecules whose function we know. That means they are actually involved in certain metabolic process. Then such metabolites, we call it as primary. The components in the body whose function is not known or which are not involved in any metabolism, then such metabolites, we call it as secondary. So the biomolecules whose function is known are called primary metabolites, and the biomolecules whose function is not known are considered as secondary metabolites. Now coming to the examples, primary metabolites, you can have the example like chlorophyll. We know the function of chlorophyll. It is a pigment, isn't it? Chlorophyll A, B, C. We have green color pigments which are involved in the uh, taking the sunlight, and then it is involved in photosynthesis. That is nothing but carbon fixation in the plants. Then amino acids involved in protein synthesis, sugars, hemoglobin, all these things. So these are the components whose function we know, and they are actually involved in metabolic processes in the body. Now, coming to the examples of secondary metabolites, we can have drugs, pigments, rubber. See, these things are useful for us. That means as a humans, we use the drugs, rubber, which we obtain from plants. But what exactly is the function of rubber in plants, that we don't know. Similarly, the drugs which are produced by the plants, we are using the drugs. But why the plants are producing the drugs? What is the function of drug in the plants? That we don't know. Clear? And if you talk about pigments also, now chlorophyll pigment we know, but there are certain pigments whose function we still don't know. So such elements or such compounds whose function we don't know, or they are not involved in any particular metabolism, then such metabolites, we call it as secondary. Now coming to a table, again very important, which is given in your textbook. So it is some secondary metabolites given in your textbook. Again, very, very important from exam point of view. So here we have a list like pigments, are carotenoids, anthocyanins. If you talk about anthocyanins, they are water soluble pigments. Sometimes they will ask you which of the following pigments are water soluble. Chlorophyll, carotenoids, xanthophylls, everything are water insoluble except for anthocyanins. So this could be an important point. Next, we have alkaloids, morphine, codeine. Okay, morphine you might have heard in human health and diseases in drugs. Then terpenoids, monoterpene, diterpene, very easy to remember. Essential oil, lemongrass oil, again very easy to remember. Okay, now toxins, lectins, drugs, polymeric substances. These are very, very important from exam point of view. Okay, so toxins, example will be heparin, ricin. Lectins are concavalin A. Now, if you ask the meaning of lectins, lectins are actually proteins. They are proteins which are bound to carbohydrates. All right, so there are certain proteins which are bound to carbohydrates or certain sugars in plants. Now, what is the function of these lectins in the plants? We don't know. And the example of lectin will be concavalin A, very important. Drugs will be vinblastine and curcumin, etc. It's curcumin. Curcumin, if you say, it is found in turmeric. See, turmeric is haldi. So if you look at the turmeric functions, we say it has got medicinal properties. Okay, and why it is having medicinal property? Because of this product called curcumin. So it is found in turmeric. Next, we have polymeric substances like rubber, gum, and cellulose. Now, here again, very, very important if you talk about cellulose. See, where do we find cellulose? It is seen in cell wall of plants. So if someone asks you what is the function of cellulose, we can easily say it is present in the cell wall. It is giving protection or it is involved in giving the structure of the cell in the plant, isn't it? So even though we know the function of cellulose, but still it comes under secondary metabolite. Very, very important. So usually I've seen students getting confused with this cellulose. They say we know the function of cellulose now. So why don't we consider it as primary metabolism? No, answer is secondary metabolism only. So cellulose will be considered as secondary metabolism only. So this table is very, very important from need point of view. Definitely one question will be coming from this table. So this is all about the introduction part of this chapter. Next, we'll talk about the carbohydrates. So here, carbohydrates are made up of carbon, hydrogen, and oxygen. These carbohydrates are divided into three types: monosaccharides, oligo, and polysaccharides. So here carbohydrates are otherwise called as saccharides. Now, what do you mean by the term saccharide? Nothing but sugars. Okay. Now, these carbohydrates are classified into three different types based on the number of the sugar units. So based on the number of sugar units, say for example, if my circle, this circle is my one sugar unit, say this is my sugar unit or one saccharide. So based on the number of sugar unit present in the carbohydrates, I can classify carbohydrates into three types: mono, oligo, and poly. So here monosaccharide means only one sugar unit. Oligo means few sugar units. Poly means many sugar units. So based on the number of sugar units, mono, oligo, and poly. Again, if you consider the monosaccharides here, monosaccharides again can be classified into different types based on the number of carbon atoms in it. So what happens here? Say this is my sugar unit. So number of sugar will be only one in monosaccharide. But this single sugar can be made up of 3 carbons, 4 carbons, 5 carbons, 6 carbons, like that. So it can be made up of different number of carbons. And based on the number of carbons, we can call it as triose, tetrose, pentose, hexose, and heptose. It is depending on the number of carbon atoms. In your syllabus, they have mentioned the examples for the pentoses and the hexose. So we'll be discussing only these two in detail. Pentose example they have given is ribose and deoxyribose. These are the two examples they have mentioned in pentose. And hexose, if I give you the example, we have fructose, then glucose, then we have galactose, then mannose. Okay, all these are the different examples for hexoses. So only these pentose and hexose will be discussing in detail later. This is about the monosaccharide. Again, coming to the oligosaccharide. Oligo, as I've told, means few. Oligo means few. So what are few here? The sugar units, what I have drawn here, the circles, are only few in number. Now, what could be the number here? It is from 2 to 9. So it is not 1 to 10, it is 2 to 9. If the number is 2 to 9, number of monosaccharides, all are bonded with glycosidic bonds. So this type of sugar or carbohydrate, we call it as oligosaccharides. In oligosaccharides, again, only disaccharides are mentioned in your syllabus. Apart from disaccharides, we also see the trisaccharides. Then we have tetrasaccharides. disaccharides. Now, what do you mean by this? Disaccharides is a condition where we can have the sugar units, which are 2 in number. So, di means 2. So, we have only 2 sugar units. If you talk about trisaccharides, we can have 3 sugar units with these bonds. Tetra will have 4 sugar units. Now here, please don't get confused between the trioses, tetroses with trisaccharide and tetrasaccharide. Many students have seen, they will be getting confused. Which of them are oligosaccharides, which are monosaccharides, whether triose is oligo or mono. Okay, so please don't get confused. Triose, please remember, triose is a monosaccharide with 3 carbons. Tetrose is again a monosaccharide with 4 carbons. Trisaccharides have got 3 sugar units. Okay, it has got 3 sugar units. Tetrasaccharide have got 4 sugar units, and these two are example of your oligosaccharides. Clear? So this is about the oligosaccharides. Next, if you talk about polysaccharides, polysaccharides are the carbohydrates where the number of monosaccharides will be more than 10. So here, if suppose these are the monosaccharide we have. So all these monosaccharides are bonded by the glycosidic bonds. And then if the number of monosaccharide is more than 10, such carbohydrates, we call it as polysaccharides. These polysaccharides again can be classified based on their composition and function, which we'll discuss later in detail. Now, coming to the first type of carbohydrates, we have monosaccharides. So as I've already told, mono here means single. So we have only one saccharide. Saccharide means sugar. So we have only one sugar unit, then we call it as monosaccharide. And here I have written they cannot be further hydrolyzed. Now, what do you mean by the term hydrolyzed or hydrolyzation? For example, we have, say these are the sugar units. If I'm taking oligo, where we have few sugar units like this, attached with certain bonds. Okay. So when you break these bonds, the bonds which are present like this, when you break these bonds by using water molecule, hydro means water. So when you are breaking the bonds by using the water molecule, then that condition, that action, we call it as hydrolysis. Breaking the bonds in the presence of water is called hydrolysis. So in case of oligosaccharides or polysaccharides, there is a chance of breaking the bonds between the sugar units. Hence, we can say oligosaccharides and poly can be undergone hydrolysis. But if you consider monosaccharide, where we have only one sugar unit, which is not attached to any other sugar unit, so there is no chance of breaking of bonds between them. We don't have any other monosaccharide. Hence, I have written that they cannot be hydrolyzed further because we have only one sugar unit. There is no bonds between the sugar units there. Okay. Now, as I have told, in monosaccharide, in your syllabus, we'll be discussing only 2 types. The first one is called pentoses and the second will be hexoses. If you talk about pentoses, what is the meaning of penta here? Penta means 5. So here we have 5 carbons. Okay, so if we have 5 carbons in one sugar unit, such sugar we call it as pentoses. Example will be ribose and deoxyribose. Structure is very important. So now we're going to learn the structure of ribose. If you talk about the ribose, as we already know, it is a 5 carbon sugar. So I'm giving you logic and trick to remember. Okay, so please try to learn where I'm writing this structure. So ribose has got 5 carbon in it. 5 is related to pentagon. So first draw the pentagon here because it is 5 carbon sugar. Okay, 5 pentagon. Now what you do, if you talk about the molecular formula for ribose or any other monosaccharide, it will be CN, H2N and ON. As I have told, carbohydrates are made up of carbon, hydrogen and oxygen. Clear? Ribose is a 5 carbon. So carbon number will be 5. So hydrogen will be 10, 2N is 10 and oxygen will be 5. So in this diagram, what I have drawn, the pentagon, I have to fill all these things. Clear? Now I have 5 oxygen. So I'll start with oxygen here, somewhere like this. C1, C2, third carbon, fourth carbon. Clear? So 4 carbons I have written in the ring. Now what about the 5th carbon? I have 5 carbons now. So where should I write the 5th carbon? That 5th carbon will be outside the ring, somewhere here. So it will be just like your flag. Clear? So this oxygen, what I have drawn, will be something like your head. Okay. The C1 and the C4 will be like your shoulders. C1 and C4 will be like your shoulders. And C4 will be like a hand which is holding your flag. So C1, C2, C3, C4, C5 will be outside the ring. Clear? Now we have to fill this carbon with their valencies. So if I fill the carbon's valencies, C1, hydrogen, ox, hydroxyl, H, OH, H, OH, and here, since C4 already has got 3 bonds, the 4th bond will be having simply H, C5 will have OH and H2. Clear? So this will be the structure of our alpha ribose. Clear? It is alpha ribose. Why alpha ribose? Alpha and beta, it basically differs at C1. Say, for example, if this is alpha ribose, at C1, H will be above, OH will be down. If I want to draw the beta ribose, the structure of beta ribose will be something like this. Again, draw a pentagon, same. Then start with oxygen here, C1, C2, C3, C4, 5th carbon outside the ring. Okay. Now, if it is beta, I'm drawing the beta ribose. So if it is beta ribose, what happens at C1 in alpha, if you see, we have H above, OH down. In beta, OH will be above, H will be down, and rest everything will be same. Everything will be same like this. Clear? So this will be called as beta. So the change will be only at C1, H above, OH down, alpha ribose, OH above, H down will be beta ribose. Now, the next example of the pentose sugar is our deoxyribose. Now here, if you look at the term deoxy, D means removal of, oxy is oxygen. So basically, we are going to remove one oxygen from ribose. Then we call it as deoxyribose. Now, ribose structure we have already discussed here. Clear? And we have so many oxygens. So from where we have to remove that oxygen, we are going to see now. So let me draw the ribose structure again. Oxygen, C1, C2, C3, C4, and the 5th carbon outside. And now fill all the valencies here. H, OH, H, OH, H, OH, H, OH, and H2. Okay. So here I have drawn the structure of a alpha ribose. But I have to draw the deoxy. That means I have to remove the oxygen. And from where I have to remove that oxygen? It should be always from C2. So please remember, we have to remove the oxygen from C2 only. Remove the oxygen from 2nd carbon only. You should not remove oxygen from 1st or 3rd carbon or 5th carbon. Always you have to remove from 2nd carbon only. Only then we call it as deoxyribose. So these are the 2 examples of pentoses, ribose and deoxyribose. Now, where do we study this ribose and deoxyribose? The ribose, if I write down here, beta ribose especially. Beta ribose is seen in RNA, which is a type of nucleic acid, and the same way beta deoxyribose will be seen in the DNA. So please try to remember the structure of both ribose as well as deoxyribose. Clear? Now, the next example of the monosaccharide is hexoses. Hexoses, as we have already discussed, it has got 6 carbons. Okay, a monosaccharide with 6 carbons. Hexoses can be of 2 types based on their functional group. If aldehyde is present, which is nothing but CHO. If CHO is present, then such hexoses we called as aldoses. Example will be glucose, galactose, mannose. If the ketone is a functional group, which is nothing but CO, then it will be ketoses. Example will be fructose. So these are the different types of hexoses we have, but in your syllabus, they have mentioned the structure of only glucose. So glucose we are going to study now. Glucose is otherwise called as grape sugar. And in glucose again, we can have 2 types, alpha glucose and beta glucose, the same thing what we have discussed for the ribose. Now, coming to the structure of glucose, first I'll draw the alpha glucose. So again here, please try to remember the logic. Glucose has got 6 carbons. So first try to draw a hexagon with 6 sides. And here, you start again with oxygen. Now 6 carbon, Cn, H2n, ON. Okay. So C6, H12, O6. Now here again, start with oxygen, C1, C2, C3, C4, C5, and the 6th carbon will be outside the ring, just like your ribose. Again, fill the valencies here. So C1, H, OH. Oh, please remember C1, if H is above, OH is down, that will be alpha. OH. One more point you have to remember is any glucose, whether it is alpha or beta, at C3, OH will be above, H will be down. Okay. C4 again, same thing. C5 already 3 bonds are there. So we have only H, OH, and H2. So this is the structure of our alpha glucose. Clear? If you talk about beta glucose, again, draw a hexagon. Since it has got 6 carbons, start with oxygen. This side, C1, C2, C3, C4, C5, and the 6th carbon will be outside the ring. Again, fill the valencies. So here, now again, beta. So here beta glucose, as I've told, alpha and beta differs at only C1. In alpha, H and OH are like this. In beta, OH will be above, H will be down. So this will be beta. Rest everything will be same. OH. C3 again, you have to remember OH will be above always. Then H, OH, H here, then OH and H2. Okay. So these are the diagrams or the structure for alpha and beta glucose you have to remember. Now, if you look at the NCERT, these diagrams are given in the textbook. So out of these two, ribose and glucose. Okay. Ribose, we have 5 carbons. So draw pentagon, oxygen, then C1, C2, C3, C4, and the 5th carbon is outside. And again, fill their valencies, H, OH, H, OH, H, OH. Here it will be H, CH2 and OH. Clear? So this is alpha ribose because H is above, OH is down. So this is your alpha ribose. On the other hand, if you talk about glucose, glucose has got 6 carbons. So draw a hexagon. Start with oxygen here. C1, C2, C3, C4, C5. And this is your 6th carbon. Okay. And again, since this is alpha glucose, so H and OH, H and OH. And you have to remember at C3, OH will be above, H will be down. OH, H, and this. Okay. So this is the diagram you have to remember since it is given in your textbook. Again, very important. We can expect in the exam. Now, this is all about the structures of your monosaccharide, pentoses, and hexoses. Next, we'll talk about something called derived monosaccharides. Now, this is not given in your textbook, but you can remember if you want. Now, what do you mean by derived? These are nothing but monosaccharides slightly modified. For example, we have deoxy sugars. We have a sugar that is carbohydrate in which we have removed the oxygen. Now this we have seen deoxyribose. Okay, we have a ribose sugar, proper ribose sugar, wherein we have removed the oxygen molecule, it becomes deoxyribose. Next, if you add an amine group is NH2. So if you add an amine group to a sugar, it becomes glucosamine. If you add an acid, carboxylic acid, to a sugar, it becomes a sugar acid, glucuronic acid, ascorbic acid. Okay, so here sugars are slightly modified by adding an additional functional group. On the other hand, if you add alcohol, that is OH, to any sugar, that becomes sugar alcohols. Example is mannitol, very important. Mannitol is obtained from brown alga. This you will study in your plant kingdom. Okay. So these are the different modified versions of monosaccharides. With this, we have finished monosaccharides. Next, we'll talk about oligosaccharides. Coming to oligosaccharides, oligo again means few. Saccharides are the sugar units. So if you have few sugar units, then what are the number? I told 2 to 10, or we can say 2 to 9. If 2 to 9 is the number, we call it as oligosaccharides. And what are the different types of oligosaccharides? Based on number of sugar units, we can have trisaccharides, and tetrasaccharides. Please don't get confused between trioses and trisaccharides, what I have mentioned before. Now again, disaccharides is mentioned in your syllabus, so this we'll discuss. Coming to disaccharides, if there are two sugar units which are bonded by a glycosidic bond, then such sugars we call it as disaccharides. Example, sucrose, maltose, and lactose. Now here you have to remember the components of which these disaccharides are made up of. In all the three, glucose is common. So if I write down glucose, here also glucose, here also glucose. All right. So here, if this is the glucose, now we have to find out what is the other component, other sugar of which it is made up of. So first, sucrose, it is glucose plus fructose. Then maltose, glucose plus glucose. If it is lactose, we have glucose plus galactose. Okay. So for this, again, I can give you one trick because many people get confused in the exam. If you talk about sucrose, so sucrose we usually get from sugarcane. Okay, till your NEET exam, just remember sugarcane, even though it is a stem, modified stem, you remember it as a fruit, only for biomolecules chapter. Okay, so sugarcane is a fruit, and from this fruit we are getting sucrose. So glucose plus fructose. Fructose usually is seen in fruits, is it? So for that, this trick will be useful. Then, if you talk about maltose, if you have heard that Horlicks is made up of malt. Have you heard? Horlicks is made up of malt. Malt is nothing but your maltose. What happens when you drink Horlicks? We get lots and lots of energy, okay, isn't it? And this energy usually we get from glucose. So since the Horlicks have got lots and lots of glucose, we get energy. Try to remember like that. And this Horlicks have got maltose in it. So maltose has got all the glucose components only. So glucose plus glucose gives maltose. Clear? And then we are left with lactose. Easy to remember. Galactose, lactose. Here also galactose. Okay. And lactose plus glucose. Glucose is common for all the three. You have to remember the second sugar for the disaccharides, which is mentioned in your textbook. So these are the three examples of disaccharides, very, very important from NEET point of view. Apart from disaccharides, we have trisaccharides, tetrasaccharides. Now, these are not mentioned in your textbook, but for extra information, you can remember the example raffinose, is trisaccharide. Raffinose, if you want to remember, you can say it is glucose plus fructose, whatever you know, and galactose. Okay. And then if you talk about tetrasaccharide, it is stachyose. Components not necessary. So this is all about your oligosaccharides. Next, we'll be talking about the bond which is present between the monomers in the carbohydrates. So that bond is called glycosidic bond. Now we'll see how this glycosidic bond is formed. So for this, I'll take an example of two sugar units, that is two glucose units. I'll take alpha glucose and then we'll try to make a glycosidic bond. So in order to make alpha glucose, now you can draw along with me the alpha glucose structure. So here we have 6 carbons. So draw hexagon. Okay. Oxygen, C1, C2, C3, C4, C5, 6th carbon outside the ring. And then you fill all these with the valencies. H, OH, H, OH, C3, OH will be above, H will be down. OH, H, OH, and H2. Okay, this is one alpha glucose. Let me draw the second alpha glucose again. Say this is my next molecule. So here also oxygen, C1, same thing. C5, C6, and fill the valencies here. OH, H, OH, OH, and H, H, OH, H, OH, and H2. Okay. So I have taken two alpha glucose molecules, and now we are going to see how do we get or how are these bonded with each other. So whenever there is a bond formation, we have to remove one water molecule from this. And from where exactly you are going to remove that water molecule is the OH of C1 and the H of C4 of the second sugar unit, you will be removing in the form of water. So what are we left with here? If you observe, we are left with oxygen, isn't it? So one oxygen will be bonding between the two alpha glucose. So if I draw this again, I can show you clearly. So if I draw this, not the complete diagram, let me draw at least the structure. So this is C1, oxygen, C2, C3, C4, C5, and the 6th carbon outside. Similarly, oxygen, C1, C2, C3, C4, C5, and C6. Okay. Now here, if you see, I have removed OH of C1 and H of C4, and it is removed in the form of water molecule. And what we are left with here is the oxygen. Now this oxygen will be binding the two alpha glucose units. Okay. And this bond, what we get is our glycosidic bond. So glycosidic bonds are present between the carbohydrates between the monosaccharides of the carbohydrates. Now, if I want to name this bond, that means in between what carbons it is present. So here, since I have taken alpha glucose unit, so let me write down the alpha first. C1 here and C4 here. So it is present between 1, 4. Okay, alpha glucose, C1 and C4, glycosidic bond. So this is how we name the bond between the two sugar units in carbohydrates. I hope it is clear to everyone. So this is all about the formation of glycosidic bonds in the carbohydrates. Next, we will be talking about something called reducing sugars and non-reducing sugars. Again, this is very, very important point. Reducing sugars if I tell you examples, let me tell you first. Examples will be your glucose, then fructose, then we have lactose. Next, we have the maltose. Okay, etc. We have so many others. So these are the sugars, we call it as reducing. Now, why we call them as reducing are, in these sugars, whenever they are formed or when they have got free aldehyde or ketone groups in their structure. Now, what does this aldehyde and ketone group do? They will reduce the cupric ions. That means the Cu plus 2 to Cu plus. So, for example, we have certain solution called Benedict's solution or Fehling's solution. So, in these solutions, we have the copper solution, which is present in the form of cupric ions. Now, cupric ions will be Cu plus 2. Now, when these sugar molecules are added to the Benedict solution, these sugars will convert cupric ions to cuprous. So basically, they are reducing cupric ions to cuprous ions. Hence, we call such sugars as reducing sugars. So where do you use this term as reducing and non-reducing, or when do you use this Benedict's and Fehling's is whenever we do the sugar test, that is the blood glucose levels or the urine test to detect the sugars in our body, we use the Benedict solution or the Fehling's solution in that test to detect the blood sugar levels or the urine sugar levels. So in that Benedict's or Fehling's solution, we have cupric ions. And whenever we have this type of glucose molecules, sugar molecules in our body, these sugar units will convert they will reduce cupric ions to cuprous ions because they have free aldehyde or ketone groups in their structure. Hence, these sugars are called reducing sugars. And the only sugar in your syllabus which is mentioned is sucrose, which is considered as the non-reducing sugar. Very, very important from NEET point of view. So sucrose is a non-reducing sugar because this sucrose do not have free aldehyde or ketone group to reduce cupric ions to cupric. It cannot do it. Hence, sucrose, which is made up of glucose plus glucose. Okay. So this sucrose cannot reduce cupric to cuprous. Hence, it is called as non-reducing sugars. Again, very, very important for the exam. So this is all about our oligosaccharides. And then we will talk about something called polysaccharides. Polysaccharides, as we have already discussed, poly means many, or we can say chain of monomers, that is saccharides. And they are macromolecules, biomacromolecules. We find them in retentate part or acid insoluble part of the chemical analysis. Okay. Now, if you consider the structure, how do these polymers appear? They are thread-like structure, just like your cotton. Okay, so they are thread-like structures. Now, these polysaccharides, when all the glucose molecules or all the monosaccharides are arranged one after the other, they can be branched or they can be unbranched. Now, here very important branched examples are glycogen and starch. These are the examples of branched. Unbranched examples will be cellulose, chitin, peptidoglycan. Okay. So these are the examples for unbranched polysaccharides. Next, very, very important point. If you talk about a polysaccharide wherein we have so many sugar units which are bonded by the glycosidic bonds like this, say these are the sugar units bonded by the glycosidic bonds. Now this side is your left hand side and this is the right hand side of the polysaccharide. The right hand side of the polysaccharide is the reducing end. This is very important. They are having the reducing end. That means they have free aldehyde or ketone groups. They can convert cupric ions into cuprous. And here, the left hand side, they are always non-reducing end. So this is very important. In a chain of polysaccharide, right hand side, it is reducing. You can remember R with R. Right hand reducing and the opposite, that is left hand, will be your non-reducing. Very important. And the last point, they are usually insoluble. So polysaccharides, cotton, if you see, or we have wood, paper, right? All these things are not soluble in water. So they are usually insoluble in water. Next, we'll classify these polysaccharides into different categories based on, the first classification is based on composition of which molecules or which components they are made up of. So if you consider the composition, polysaccharides can be of two types: homopolysaccharides and heteropolysaccharide. Homo means same. So if all the monomers or the monosaccharides are of same type, we call it as homopolymers or homopolysaccharide. And if we have different types of monosaccharides, then such polysaccharides are called heteropolysaccharides. Examples of homopolysaccharides will be many, like glycogen, starch, then we have inulin, next we have chitin, cellulose. All these are the examples of homopolysaccharides, wherein they can be made up of glucose, NAG, all this. So we can have different types of monosaccharides also. Now first coming to the structure of glycogen. Glycogen is made up of alpha glucose units. So it is a polymer made up of alpha glucose only. So only alpha glucose are present. But this alpha glucose are arranged in different forms. For example, we have a main stem. For example, this is the main structure, or let me draw differently. Say these are the sugar units here, which are bonded by alpha 1,4 glycosidic bonds. Okay. And now from these main stem arises the branches. So let us say from this glucose, one more branch is arising like this. So from here, like this, and again like this. So it goes on. So we can have the branches over the main stem. And you have to remember that all these branches, whatever we have, those are linear. So we have a main branch or main stem, which have got alpha 1,4 glycosidic bonds. And wherever these branches are arising, for example, at this point or this point, here we will have alpha 1,6 glycosidic bond. That means the bonds will be present between first and the sixth carbon. So this point you have to remember. If you look at this diagram, what I have drawn. Now this orange one, what I'm drawing now, is your main stem, which have alpha 1,4 glycosidic bond in it. And the point from where the branches are arising, let us say this part and, this part. So these points from where the branches are arising, they will be made up of alpha 1,6 glycosidic bond. This is the structure of glycogen. Next, if you talk about starch. Now, starch is also made up of alpha glucose units. And here, glucose is arranged in the form of two components. We have something called amylose, which forms the 20% of the starch, and amylopectin, which forms 80% of the starch. So these are the two components which we see in starch. And please remember, both the components are made up of alpha glucose units only. So here, now how are these arranged? If you talk about the amylose, amylose is arranged in the form of helix, something like this. And on this helix, now this helix is again made up of alpha 1,4 glycosidic bonds. So whatever glucose units are there, that will have alpha 1,4 glycosidic bonds again. And the site from where the branches are arising, like this. Like this. So this is the site where the branches are arising. Again, at this site, we have alpha 1,6 glycosidic bonds. And please remember, the amylose component will be the helical. Now this is amylose component, which is helical, whereas amylopectin will be linear. Whereas if you consider the structure of glycogen, we don't have different components in glycogen. Everything is made up of alpha glucose unit. But here we have a main stem from which branches are arising. Everything is linear here. Everything is linear and branched. Whereas in case of starch, amylose will be helical. This is helical. Amylopectin will be linear. But if you put together, both of them are branched. So glycogen and starch are branched polysaccharides. Next, if you talk about inulin. Inulin, chitin, cellulose, all these structures are not given in your textbook, but you have to remember what is the component it is made up of. So if you talk about inulin, it is a polymer, or we can say homopolymer of fructose. So till now, glycogen and starch, we have seen that is a homopolymer of glucose, whereas inulin is a homopolymer of fructose, very, very important. And where do we find this inulin? It is found in roots of a plant called Dahlia. 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 Here. And here, please don't get confused between inulin and insulin. People usually try to confuse you. Inulin, please remember, it is a homopolymer of carbohydrates, whereas insulin is a heteropolymer of protein. See, proteins are always heteropolymer because the proteins are made up of different types of amino acids. Not a single protein we have which is made up of all the same type of amino acids. Okay. Say, for example, this is your protein structure. Each circle, if I denote it with amino acid, let us say this is made up of methionine, this is made up of cysteine, this is phenylalanine, okay, this is tyrosine. So all these are made up of different amino acids. Hence, proteins are always heteropolymers. So here I can say insulin is always heteropolymer, whereas inulin is a homopolymer. One more point, insulin is a protein, inulin is a carbohydrate. So these points you have to remember with respect to inulin. Next, coming to chitin. Chitin is again a homopolymer of NAG. What do you mean by NAG? N-acetyl glucose, N-acetyl glucose amine. So chitin is formed by only NAG. So we have NAG, which are again joined by glycosidic bonds. The last one, cellulose. We find this in cell wall of plants. This is made up of beta glucose. Again, this is important. Starch and glycogen is made up of alpha glucose. Cellulose is made up of beta glucose. Now, where do we see this cellulose? We can see in paper, cotton, wood. So these are the examples where we can find the cellulose. All right. Now, these are all the examples of our homopolysaccharides. Next one, if you talk about the heteropolysaccharide, example is peptidoglycan. Peptidoglycan is found in the cell wall of bacteria that we'll discuss later. Before that, if you talk about the structure, again, in detail structure is not necessary. You have to remember only the component. It is made up of NAG plus NAM. Now, what is NAG? Just now we have mentioned perchitin, N-acetyl glucosamine. So here, NAG will be N-acetyl glucosamine. N-acetyl glucosamine plus NAM, N-acetyl muramic acid. So these are the two different types of monosaccharides of which the peptidoglycan is made up. So this is the first classification of polysaccharides based on its composition. Coming to the next classification of polysaccharides based on the function. What kind of function the polysaccharides are performing? Now this will be, so it is not the homo. It is based on the storage and structural. That means whether the polysaccharide is involved in storing the components or the monomers, or whether it is involved in providing structure to any component. So example of storage polysaccharides will be glycogen, starch, and inulin. If we talk about glycogen, here glucose is stored in the form of glycogen in animals. Isn't it? In animals, for example, humans, any other mammals, or any animal, if you consider, the glucose is stored in the form of glycogen. And where is it stored? In liver and muscles, in case of humans, if you talk. Clear? Same way, starch. If you consider, here also glucose is stored in plants in the form of starch. And please try to recollect the structure of glycogen. Glycogen is branched. Both glycogen and starch are branched. Glycogen will have a linear branch like this, whereas starch will have amylose like this and then amylopectin. So it has helical structure as well as the branched, which is amylopectin. Now, one more point in starch I wanted to tell you is, if you consider the starch and the glycogen, both of you, if you treat with iodine, treated with iodine. So when you treat the starch with iodine, if you remember, you have done the experiment of potato. When iodine is placed on potato, the potato will turn into bluish black color, isn't it? Now, why is that happening? Because the potato has got starch in it. The iodine, when you put the iodine on the potato, this iodine will get trapped in this helical structure of the amylose. Now, this is amylose, which is helical. So in this, the circles, the helices of this amylose will trap the iodine molecules. And since the iodine molecules are getting trapped in the amylose helices, the entire starch which is present in the potato will appear blue-black color, because iodine itself is a blue-black color. Whereas the glycogen, just now we have discussed, it does not have any helices. It is a linear structure. So iodine cannot get bind with the, or it is not getting trapped with the glycogen structure. So hence glycogen will not show blue-black color, whereas starch when treated with iodine shows blue-black color. So this is again very important. It is only because of the helices which we see in the starch. Okay. Now coming to inulin. Inulin is also involved in storing of fructose. So just now I mentioned before, fructose, the monomers, that is fructose, will be polymerized to form inulin in the roots of dahlia. Now, if you talk about the importance of inulin, inulin is used to test the kidney function. So it is basically used in kidney function test. So what do you mean by this kidney function test? If any person is suffering from kidney disorders, so whether the kidneys are functioning or not, that will be tested with inulin. So when we take inulin in our diet, this inulin is not getting metabolized. That means it is not used anywhere in our body. It will directly enter in your kidneys along with the water, and from the kidneys, it will come out in the form of urine. So along with the urine, the inulin will come out. So when you detect the rate of urine formation, in that urine formation, the amount of inulin which we get will tell you how is the function of the kidneys. Clear? So this is the function of inulin when in the kidney function test. So these are the three polysaccharides: glycogen, starch, and inulin, which will be acting as storage polysaccharides. Next, we will talk about the structural polysaccharides. The structural polysaccharides will give the structure to any components. For example, peptidoglycan, if you consider here, this will give structure to the cell wall of bacteria. All right. And we know peptidoglycan is made up of NAG plus NAM. They are heteropolymers. Next, coming to cellulose and hemicellulose, they will be giving the structure to the cell wall of plants. Next, chitin, they are again giving the structure to the cell wall of fungus. Apart from this, they also give structure to the exoskeleton of arthropods. So these are the functions of chitin. And next, pectin, if you consider, they are again involved in the cell wall of plants. All right, so these are the different types of polysaccharides involved in giving the structure. And if you see now, again in the exam, they will ask you which of the following polysaccharides are branched or unbranched. So I'll give you a hint or a logic for this to understand. In your textbook, they have mentioned that glycogen and starch are branched, whereas peptidoglycan, cellulose, chitin, they are unbranched. So let me write down those points and then I'll tell you the trick. So glycogen, starch are the branched polysaccharides, whereas peptidoglycan, chitin, cellulose. Only these points are mentioned. So based on this, I am telling you the trick. So these are unbranched. Only these options will be given in your exam. Now, if you consider the functions, glycogen and starch, they are involved in storage. Okay, they are involved in storing the glucose. Glycogen in animals, starch in plants. Whereas all these things are involved in providing the structure in the form of cell wall. Isn't it? Now, if you consider a wall, any walls in your rooms or in your house, whatever walls we have, are the walls branched? Can a wall be branched? Answer is no. Walls which are present in our rooms, they are not branched, isn't it? So just remember, whatever polysaccharides are involved in giving structure, clear? Involved in structure like peptidoglycan, chitin, cellulose, they are basically involved in structure, that is cell wall. They are forming the walls, cell walls. Hence, they are unbranched. Now, this is not the reason. I'm telling you the logic to remember. Yeah. Whereas the storage polysaccharides, that is glycogen and starch, they will be branched. Now, don't ask me about inulin. Don't try to remember the inulin. Here, branched, unbranched, you have to remember only with this respect to these 5 elements only. Okay. And the last point with respect to this classification, as I've told, glycogen and starch are the storage polysaccharides in animals and plants respectively. So here glycogen is stored in, the glucose is stored in animals, and in plants, glucose is stored in the form of starch. So my question here is, why can't we store glucose as it is in the animals as well as plants? Why this glucose needs to get converted into glycogen and starch for their storage? If you remember, like whenever we want to have the energy, the respiration, for energy, we have to have the glucose. Clear? So we take food. This food will be broken down into glucose. This glucose will be converted into glycogen. And again, whenever we want energy, this glycogen will be broken down into glucose again. And then we get what? Energy. Clear? So this glucose, which was formed as a result of digestion, is stored in glycogen, and then again it is getting converted into glucose whenever we want energy. Why can't we store this glucose? Well, even though it is animals or plants, why can't the organism store glucose as it is so that it can be used as it is whenever we require the energy? Okay. So for this, the answer is, first and foremost point is glycogen and starch. Both of them are polysaccharides, isn't it? They are polysaccharides which are very, very bulky. That means they are very large and very, big molecules. So they are bulky and they are not dissolved in water. So they do not dissolve in water. I mean, where do we have water? You can ask. In the cell, this is a cell. It can be plant or animal cell. So in the cell, the carbohydrate or the glycogen or starch is present as it is without getting dissolved with the cytoplasm. So it is basically chemically non-reactive with the cytoplasm or the water which is present in the cytoplasm, and it is stored in bulk. Clear? So as a result, what happens? It is osmotically inactive. Now, what do we mean by this term, osmotically inactive? If you consider the glucose, say for example, we have a cell wherein we have glucose. Now, glucose, when you dissolve glucose in the water, it will get mixed up. I mean, it gets dissolved in the water. Same way, when the glucose is present in the cell, it will get dissolved in the cytoplasm. And this glucose will act as a solute. Now, what happens? The water which is present around this cell, okay, now we know the osmosis and everything. Usually, the water which is present around this cell will tend to enter inside the cell because of the more concentration of solute. Because whenever we have more amount of solute, any side, that side, the water tends to move in. So if the cell is storing more and more glucose, more and more water tends to enter inside the cell. As a result, the cell will, swell, and at the end it can burst also. So these are the chances of the cell when the glucose is stored as it is, because it is acting as a solute here. Okay. On the other hand, if the same glucose is getting converted into glycogen or starch and then it is stored in the cell, first point, it is not getting dissolved in the cytoplasm. Clear? Since it is not getting dissolved, it is not chemically reactive. It is not forming any solute. So it will not act as a solute. Okay, glycogen and starch is not acting as a solute. As a result, the water which is present surrounding the cell will not enter inside the cell. So this, we call it as osmotically inactive. Clear? So this is why the glucose is stored in the form of glycogen and starch in animals and plants respectively. So this is all about the carbohydrates, and we have finished the introduction part in this chapter and then the carbohydrates. Next, we have few important points in this chapter. For example, the most abundant biomolecule. See, we have 4 biomolecules given in your chapter: carbohydrates, proteins, lipids, and nucleic acids. Out of these 4, if they ask which of the following is the abundant biomolecule, so abundant biomolecule will be considered as cellulose. This is important. But if they ask the most abundant protein, now protein we didn't start yet, but still, if they ask the most abundant protein, the answer will be rubisco, which is present in plants. And this rubisco is involved in carbon fixation. Carbon fixation is nothing but photosynthesis. All right, so rubisco will be the most abundant protein in the biosphere. Protein in biosphere. And if they ask the most abundant animal protein in the biosphere, then the answer will be collagen. All right, so these 3 points are very important in this chapter. So with this, we have finished the carbohydrates and the introduction part. In the next video, I'll take up the proteins and the lipids. Thank you.